Hello everyone. In this class, we'll learn about learning and memory. The learning objectives would be define and classify learning and memory, give the types and significance of conditioned reflexes, describe the mechanism of learning and memory, name the brain areas involved in different types of learning and memory, and understand the physiological basis of abnormalities of learning and memory. Learning and memory are closely associated higher functions of human being. This is because the process of learning involves storage of new information in memory and its retrieval at appropriate time, and the process of memory involves repeated acquirement of new knowledge, that is learning. It is known that frequent learning of newer facts increases horizon of memory. Thus, learning utilizes memory and memory utilizes learning. Though learning involves memory, it is certainly more than the memory per se. Memory is the simple repetition of what has already been performed or stored, whereas learning involves more than the simple use of memory. It uses all less understood processes like reasoning, cognitive process, and common sense. The common sites of learning and memory in the human brain are the association areas of cerebral cortex and subcortical structures in the temporal lobe, including the hippocampus and amygdala. The association cortical areas imbibe sensory information from the somatosensory cortex and visual, auditory, and olfactory cortices. They also receive information about the emotional feelings from limbic system. These information are integrated with the previous experience of learned skills and are then stored in the memory. Coming to the definitions, learning is defined as acquirement of information or knowledge by experience that results in the alteration of behavior. Learning is the sincere attitude and readiness to perform an assigned task. It depends mostly on the motivation that creates adequate interest and attention to promote learning. Memory is defined as retention of learned information and experiences. The stored information should be retrieved and utilized at any time in life whenever needed. Thus, memory has four stages. That is registration of memory that includes proper perception and attention. Failure of learning and memory occurs due to the impaired perception and attention because the material to be learned is never registered and assimilated. Then integration and retention. Learned experiences are processed and integrated by various structures in central nervous system and then retained at appropriate place in the brain in the form of short-term and long-term memory. Then recognition and recall. At the appropriate time and place, the memory is recalled for the proper use. Then reutilization. Memory, the learned experience is utilized for the improvement of further uh, in learning, for further learning. There are different types of memory. Memory is broadly divided into explicit or declarative memory and implicit, which is also called as non-declarative memory. Explicit or declarative memory, that is uh, the memory of facts, that is semantic memory, and the memory of events, that is also called as episodic memory. The explicit memory can be short-term memory or long-term memory. Then it is also the classified as an implicit or declarative memory. So there are further classification in the implicit memory. They are priming, procedural memory, which are related to skill and habits, can be associative learning. In associated learning, there can be classical conditioning and operant condition. And non-associative learning can be habituation or sensitization. Coming to explicit memory, the explicit memory, also known as declarative memory, is connected with awareness. It has two forms: memories of events, that is called as episodic memory, and the memories of facts, which is called as semantic memory. The declarative memory is dependent on the hippocampus and the other parts of the medial temporal lobes of the brain for its retention. So it can be short-term memory. Short-term memory is the memory that lasts for seconds or 2 hours, during which the processing in the hippocampus and elsewhere lays down the long-term changes in synaptic strength. The working memory is a form of short-term memory that makes information available for a brief period. As a result of repeated training, short-term memory can be transferred into the long-term memory, which depends on the process called consolidation. During short-term memory, the memory traces are subject to disruption by trauma and various drugs. Long-term memory is the one that stores information for years together and sometimes for life. Long-term memory traces are remarkably resistant to disruption. This is broadly divided into explicit and implicit memory. Some forms of implicit memories also involve short-term and long-term memory. Then implicit memory. The implicit memory is not associated with awareness and is therefore called as reflexive or non-declarative memory. It includes skill, habits, priming, and conditioned reflexes. Explicit memory is initially required to develop implicit memory. For example, a learner of motorcycle riding initially remembers the steps of changing the gear. He changes gear with conscious knowledge till it becomes a reflexive habit to do so. He changes the gear then without awareness. Once the skill is acquired, the act becomes unconscious and automatic. Priming is the facilitation of recognition of words or objects by prior exposure to them. An example is improved recall of a word when presented with first few letters of it. Procedural memory includes skill and habits which once acquired become unconscious and automatic. Non-associative learning, where organism learns about a single stimulus, example are habituation and sensitization. Habituation is a simple form of learning in which repeated application of neutral stimulus elicits less and less response. The response that was first studied was gill withdrawal in aplasia when the gill is stroked. After few strokes, the response is not seen. The withdrawal is an aversive response, and if the stroke is harmless, the animal gets habituated to it. Habituation implies learning and therefore can be studied for its cellular mechanisms. It can be short-term or it can be a prolonged if exposure to the benign stimulus is repeated many times. Habituation is a classical example of non-associative learning. Sensitization is the opposite reaction in which repeated application of the stimulus evokes greater and greater response. Sensitization occurs especially when the stimulus to which habituation has developed is coupled with a pleasant or unpleasant stimulus. For example, application of noxious stimulus to gill results in greater withdrawal of the gills. That is, you see an increased responsiveness. Sensitization may occur as a transient response or if it is reinforced by additional pairing of the noxious stimulus and the initial stimulus, it can exhibit features of short-term and long-term memory. Associated learning, the organism learns about the relation of one stimulus with other. The classical example for associated learning is conditioned response. Conditioned reflexes. The reflex response to a stimulus that hardly elicited any response in the past, but presently the response to the stimulus is acquired by pairing the stimulus with another stimulus that normally produces a response is called a conditioned response. There are different types of conditioned responses. There are two types of conditioned responses: classical conditioning and operant conditioning. Conditioned reflexes have two components that are associated with emotional response and motor response. The emotional responses are regulated by amygdala and the motor responses are controlled by cerebellum. Classical conditioning. In classical conditioning, in the beginning, there is a stimulus that normally elicits a specific innate response, the response which is already present without training. The stimulus is called unconditioned stimulus. Later, an arbitrary stimulus that normally does not produce any response on application does produce a significant response when paired with unconditioned stimulus. The stimulus is called as conditioned stimulus. The typical example of classical learn conditioning is the Ian Pavlov's experiment on salivation of dog. First, Pavlov produced salivation in dog by placing a piece of meat in its mouth. Then he rang a bell just before placing meat in dog's mouth and repeated the procedure a number of times till the animal was made to salivate with the bell ringing, and finally without even placing the meat in the mouth. In this experiment, salivation in the dog by placing meat in the mouth is unconditioned stimulus and bell ringing is conditioned stimulus. Salivation in response to conditioned stimulus, sound or bell, occurred by pairing it sufficient number of times with unconditioned stimulus, that is placing the meat in the mouth. Finally, conditioned stimulus produced a response even in the absence of unconditioned stimulus, which was initially evoked only by the unconditional stimulus. There can be inhibition of conditioned reflexes. Conditioned reflexes can be inhibited in two ways, the internal and external inhibition. If the conditioned stimulus is presented indefinitely without unconditioned stimulus, the response decreases and eventually stops. This is called as internal inhibition or extinction. Conditioned reflexes response can also be abolished if the animal is disturbed externally just after the application of conditioned stimulus, and this is called as external inhibition. So there can be reinforcement of conditional reflex. It is difficult to maintain conditioned reflex indefinitely. However, if the conditioned stimulus is paired repeatedly with unconditional stimulus, from time to time the conditioned reflex becomes permanent. That is, there is a reinforcement of conditioned reflexes. Conditioned reflex can also be strongly formed by association of unconditioned stimulus with pleasant or unpleasant effect. Accordingly, there are two types of reinforcement, positive and negative reinforcement. If the unconditioned stimulus is associated with a pleasant effect, the positive reinforcement occurs, and if associated with unpleasant effect, negative reinforcement occurs. Operant conditioning. In this type of conditioning, animal is trained to carry out tasks or either to receive a reward or avoid a punishment. The unconditioned stimulus may be pleasant or unpleasant even. The conditioned stimulus is applied as a signal from in the form of light or sound that alerts the animal to perform. B.F. Skinner had extensively studied this type of conditioning, operant conditioning. The animal, usually the rat, is kept in the Skinner box in which provision is made in such a way that pressing a bar results in delivery of food pellet or prevention of an electrical shock. Initially, the response occurs by chance. However, later response occurs with a greater probability as reward follows the response. That animal learns that food is obtained by pressing the bar or uh, the shock is prevented. Thus, the reinforcement may be positive reinforcement by pressing the bar, the animal gets food, or the negative reinforcement, pressing bar, animal prevents electric shock. The conditioned motor response results in avoidance of electrical shock. Therefore, this is also called as conditioned avoidance reflex or aversion conditioning. Another example of negative reinforcement is if food aversion conditioning uh, in which the animal severely develops aversion to particular food if the taste of the food, that is unconditioned stimulus, is coupled with injection of a chemical that develops physical illness, that is conditioned stimulus. Coming to the mechanism of learning and memory. The mechanism of underlying learning and memory may be divided into cellular, it can be neural, and it can be biochemical mechanisms. Coming to molecular mechanisms, learning and memory are initiated and established by several neurochemical changes like increased synaptic connection, neurotransmitter secretion, formation of intracellular second messenger, protein synthesis, and gene activation. In sensitization and habituation, where sensitization and habituation occurs due to the change in the neurotransmitter release at the sensory neuron terminal due to the alteration in the intracellular calcium. Habituation occurs due to the decreased neurotransmitter release from the presynaptic sensory ending in response to repeated application of particular stimulus. Serotonin secretion decreases from the modulator neuron. The stimuli gradually inactive calcium channel, resulting in decreased calcium content at the presynaptic terminal that in turn inhibit neurotransmitter release. Sensitization occurs due to the prolonged action potential in the sensory endings that results in increase in intracellular calcium, which in turn increases neurotransmitter release. Serotonin release from modulator neuron has been identified to be the mediator. In sensitization, serotonin secretion is increased at the target sensory neurons. In the C. snail aplasia, the noxious stimuli causes discharge of serotonergic neurons that end on the presynaptic endings of sensory neurons. Thus, sensitization is due to the presynaptic facilitation. Serotonin activates cyclic AMP in the sensory neuron terminal. Cyclic AMP phosphorylates one of the potassium channels that closes potassium channels. This results in slowing of repolarization and prolongation of action potential. This facilitates voltage-dependent calcium influx into the sensory terminal that increases the release of transmitter by exocytosis. The short-term prolongation of sensitization is due to a calcium-mediated change in adenyl cyclase that leads to greater production of cyclic AMP. The long-term potentiation also involves a protein synthesis and growth of presynaptic and postsynaptic neurons and their connections. In conditioned reflexes, the molecular changes, in classical conditioning, pairing of unconditioned stimulus with conditioned causes biochemical changes in the target neurons. The basic mechanism involved is the prolongation of action potential that causes presynaptic facilitation. For the classic conditioned reflex to develop, it is important for unconditioned stimulus should come soon after the conditioned stimulus to cause a temporal association. The unconditioned stimulus act on neurons that are activated by conditioned stimulus. Unconditioned stimulus increase calcium in the presynaptic terminal. The long-term increase in presynaptic calcium alters adenyl cyclase activity. Thus, when conditioned stimulus activate the presynaptic neuron, adenyl cyclase is activated to greater extent that forms more and more cyclic AMP. Increased cyclic AMP causes phosphorylation of a set of potassium channels and closes the channel. This slows the repolarization and prolongs the action potential. Post-tetanic potentiation. This is the production of enhanced postsynaptic potential in response to stimulation. This enhancement lasts for up to 60 seconds and occurs after a brief train of stimuli in the presynaptic neuron. The tetanizing stimulation causes calcium to accumulate in the presynaptic neuron to such a degree that the intracellular binding sites keep the cytoplasmic calcium low or overwhelmed. The relation between learning and long-term potentiation. The physiological process of learning is inadequately explained due to the complex mechanism involved in it. A repeated stimulation of presynaptic neurons results in change in the excitability of postsynaptic neurons by altering the rate of discharge, a new protein synthesis, and neurotransmitter release. The changes are found to be associated with learning. The long-term potentiation, which is called as LTP, is an important process for establishment of learning and memory. Unlike post-tetanic potentiation, it is initiated by increasing the intracellular calcium in the postsynaptic rather than the presynaptic neuron. Increased excitability and change in the intracellular protein formation and by repeated synaptic stimulation are known mechanisms of long-term potentiation, that is LTP. In long-term potentiation, initial step is the phosphorylation of number of proteins that are stimulated by formation of receptor-mediated second messengers. Phosphorylation of proteins activates various intracellular enzymes and alters neuronal excitability. In the later stage of uh, LTP, the synaptic connections between neurons increase. The LTP occurs in the hippocampus in mammals, that is long-term potentiation. This is a process of potentiation of impulse transmission in neuronal pathways in hippocampus that lasts for days to weeks when they are stimulated at high frequency. The potentiation is mediated by calcium influx. Hippocampal long-term potentiation. Hippocampal LTP is of two types, the mossy fiber LTP and the Schaffer's collateral LTP. The mossy fiber LTP is a NMDA dependent. It is mediated by presynaptic transmission that involves cyclic AMP and IH, a hyperpolarization activated cation channel. In Schaffer collateral LTP, that is long-term potentiation, is initiated by increased intracellular calcium in the postsynaptic neuron and depends on NMDA receptors. Increased calcium level makes glutamate receptors accessible to glutamate molecules. In the amygdala, the NMDA dependent LTP is produced in GABAergic neurons. Long-term depression. Though the long-term depression was first described in the hippocampus, it was subsequently demonstrated in all the fibers as for LTP. The LTD is just opposite of LTP, that is long-term depression. It is mainly characterized by a decrease in synaptic strength. It is demonstrated by slower stimulation of presynaptic neurons, and there is a smaller rise in intracellular calcium compared to that as occurs in long-term potentiation. In cerebellum, LTD requires phosphorylation of the GluR2 subunit of AMPA, that is alpha amino 3 hydroxy 5 methyl isoxazole 4 propionic acid receptors, which may be involved in motor learning. Coming to the neural mechanisms, the brain regions which are involved are the three brain regions that have been thoroughly studied in the experimental animals, especially in the monkeys and rat, to understand learning and memory. So these three important areas are prefrontal cortex, inferotemporal cortex, and the hippocampus. Prefrontal cortex. The removal of frontal lobes in monkey resulted in delayed responses to different memory tasks. It was suggested that spatial short-term memory resides in the frontal lobe. It was further investigated by ablation studies that the spatial short-term memory is the function of dorsolateral frontal cortex. The thalamic fibers concerned with memory project to the prefrontal cortex, and from there the basal forebrain. From the basal forebrain, the fibers project to amygdala, hippocampus, and neocortex. These fibers are mainly cholinergic in nature. Inferotemporal cortex. The lesion of this part of the cortex interferes with visual discrimination, where the tactile, auditory, and olfactory cues remain unaffected. The integrity of prefrontal and inferotemporal cortices is required for performance tasks that are relatively difficult. Thus, it appears that the neural substrate for learning are task specific. The another structure is hippocampus. Hippocampus is an important component of the Pepe circuit, which is extensively connected with hypothalamus, thalamus, amygdala, and septum. The combined lesions of the hippocampus and the amygdala produce significant amnesia than the individual lesions. The neural basis of learning involves substrate of a reward. The hippocampus and the medial forebrain bundle are important structures of the reward system. Prefrontal cortex, a seat of working memory, is connected with the hippocampus and parahippocampal portion of medial temporal lobe. Bilateral destruction of ventral hippocampus in human causes striking deficit in short-term memory. They have intact working memory and remote memory. They are capable of learning new tasks and retaining pre-lesion remote memories. However, they cannot form a new long-term memory. Hippocampal connections with mammillary body, that is hypothalamus, amygdala, and thalamus are also involved in the memory. The lesion of memory body or thalamus causes impairment of recent memory. Hippocampal connection with amygdala is concerned with emotion related to memory. By MRI and evoked potential studies, recently it has been observed in humans that activity in left parahippocampal cortex and left frontal lobe increases when they recall words, and activity in right parahippocampal cortex and right frontal lobe increases when they recall pictures. New neurons are formed in the hippocampus in response to learning and memory. Neural mechanisms of declarative and procedural memory. The declarative memory refers to the memory of events and facts and the ability to knowingly access them. The declarative memory is integrated in medial portion of the temporal lobe. Patients who have undergone bilateral medial temporal lobectomy, for example, for the treatment of intractable temporal lobe epilepsy, lose their declarative memories or become incapable of forming new declarative memory, but retain procedural memory. Procedural memory refers to ability to learn and remember new skills and procedures. The procedural memory is integrated in different parts of the brain depending on the type of task learned and remembered. The learning and memory, uh, the learning and remembering new motor skill and habits require the striatum, motor cortex, and cerebellum. Remembering emotional components associated with the task and skills require amygdala. Learning the conditioned reflexes requires cerebellum and cortex. The medial temporal lobe is not involved in procedural memory. The neural mechanism of short-term and long-term memory. The short-term memory. The declarative memory is divided into short-term and long-term memory. The short-term memory is that which can be recalled for seconds to minutes, and long-term memory is that which can be recalled for days to years. Learned experiences that are newly and recently acquired can be easily recalled using short-term memory. For example, before an individual dials a telephonic number, first he sees the number and repeats uh, that mentally till the number is dialed, and then he forgets the number quickly once he starts talking on the phone. So this is a form of working memory. However, if the number is repeatedly used for uh, or is an important number, the number is stored in the memory for longer duration. Thus, permanent storage of information is based on its importance or its repeated use or on whether it is associated with an important emotional event. For memory to become more permanent, processing occurs in subcortical areas that mainly involve hippocampus. The working memory makes the information available for a brief period. The center for working memory is prefrontal cortex. Working memory has two components: verbal component that retains the verbal memory and visuospatial component that retains visual and spatial aspects of objects, that is spatial short-term memory. Coming to long-term memory, the short-term memory is converted into long-term memory by mainly three ways. By repeating process of learning frequently. Second, by adding more that one sensory modality to process of learning, for example writing, and at the same time also hearing a newly acquired knowledge. And third, by associating the process of a particular learning with a meaningful emotional event. The process of permanent storage of memory is called as consolidation. Hippocampus plays an important role in consolidating memory, which is reinforced by an emotional state that is associated with learning or experience. The medial temporal lobe is important for long-term declarative memory formation, especially the hippocampal and parahippocampal cortices. However, hippocampus is not required for subsequent retrieval of long-term memory. Long-term memories are stored in the various parts of the neocortex. Various components of long-term memories reside in concerned cortical regions. For example, visual and auditory parts of memories are located in visual and auditory cortex respectively. Therefore, once long-term memories are established, they can be easily recalled by association with similar events, that is, visual, auditory, olfactory, or somatosensory later in life. Intercortical transfer of memory. The memory is transferred from one cortical hemisphere to the other through corpus callosum, that is through the commissural fibers, anterior and posterior. If these fibers are sectioned, then no memory transfer occurs. If animal is shown and taught some task with one eye, that is the other eye being blindfolded, and then the blindfold is transferred to the other eye, the animal remembers the task and can perform it. But if the optic chiasma or corpus callosum is cut, it cannot perform the task as it cannot remember what has been learned with other eye. So this is called as split brain animal. Neural mechanisms of learning. The prefrontal cortex is critical for coordinating the process of learning and memory. The cerebral cortex processes information related to learning and communicates them to the limbic structures. The prefrontal cortex gathers sensory information from the somatosensory, visual, and auditory cortices. Prefrontal cortex integrates inputs related to language and mathematical ability in the light of previously acquired learning. The prefrontal cortex is considered as a site of working memory. The new experiences are processed in the prefrontal cortex. The processed information is then transmitted to the hippocampus. Consolidation of information that occurs in hippocampus over several hours into lasting form. Then the learned experience is stored in the association cortices from where it can be retrieved whenever needed. Role of cholinergic neurons in the memory. Acetylcholine is the major neurotransmitter in learning and memory and other cognitive function. Cholinergic neurons that are present in plenty in basal forebrain of region project heavily to the hippocampus and different parts of the cerebral cortex. The cell bodies of these cholinergic neurons are highly concentrated in the basal forebrain nuclei, especially in the nucleus basalis of Meyer-Net and the nucleus accumbens. The cholinergic fibers are also main projecting neurons from the brainstem reticular formation to the thalamus and spinal cord. More than 90% of the projection from the brainstem to the thalamic nuclei are cholinergic. Loss of cortical and subcortical cholinergic neurons, especially in the basal forebrain regions associated with dementia, and impairment of memory, abstract thinking, and judgment. The brain areas for integration of various forms of memory are given as follows. For explicit and declarative memory, uh, it is the medial temporal lobe. For short-term memory, it is hippocampus. For working, it is prefrontal cortex, and long-term, it is various parts of the neocortex. For implicit or non-declarative memory, the priming occurs in neocortex, procedural memory in the striatum, associative learning occurs at which are mainly associated with emotional responses occurs in amygdala, and with skeletal musculature they occurs in the cerebellum. With the non-associative memory, which are reflex pathways, habituation, and sensitization. Then biochemical basis of learning and memory. Changes underlying learning is repeated transmission of impulses along neural circuits that results in permanent changes in the concerned neurons. One important consequence is a new protein formation in the nerve cells. Increased RNA synthesis in response to learning has been well documented. Recently, it has been suggested that activation of a specific gene is responsible for learning. Strangeness and familiarity. Stimulation of some parts of the temporal lobe occurs uh, lobe causes the change in the interpretation of one's surrounding, such as the subject feels strange in familiar places or familiarity with the new events. Such of strangeness or familiarity helps the normal individual to adjust with the different environments. But inappropriate feeling of familiarity with the new events or surrounding is clinically known as deja vu phenomenon, a French word which means already seen. This phenomenon may occur in normal individuals. However, this usually occurs as an aura, and that precedes the onset of temporal lobe epilepsy. Some of the applied aspects related to learning and memory. Amnesia means impairment of memory. There are two forms of amnesia, that is retrograde amnesia and anterograde amnesia. Retrograde amnesia is a loss of memory for events that just precede the head injury or the disease. Is called as It is called as retrograde amnesia. Usually, the loss occurs only for short-term memory, and retrograde amnesia occurs commonly in head injury, in which the patient develops concussion. Anterograde amnesia, the inability to call the memory or to form new memories after the event, that is head injury, mental shock, or disease, is called as anterograde amnesia. Anterograde amnesia also follows head injury, but the duration covered by amnesia is usually shortens with the time. Dr. Brenda Milner, who observed a patient of Dr. Wilder Penfield in mid-50s, first reported anterograde amnesia. The patient had undergone bilateral medial temporal lobectomy for the treatment of severe epilepsy, and after the surgery, he was unable to form any new declarative memory. Dr. Milner was quite surprised to observe that the patient could learn difficult tasks, perform better with repeated trials, and retain the skill over time. However, he could not remember ever having performed the task before. The different areas of cortex are responsible for learning and memory. Therefore, the degree of amnesic deficit is proportional to the amount of cortex removed in the injury or involved in the disease process, and the nature of amnesia depends on the part of the cortex involved. This indicates that importance of cortex in learning and memory. Cholinergic synapses are mainly involved in the process. Therefore, amnesia can be produced in animals by injecting synaptic depolarizing blocker or disopropyl fluorophosphates anticholinesterase drug. Dementia is a syndrome consisting of several intellectual inabilities. The deficits occur for many cognitive functions, including learning and memory. It occurs in many conditions that affect the cortical functions. The commonest is senile dementia. The pathological dementia is commonly seen in neurodegenerative diseases like Alzheimer's disease. However, drug induced dementia and alcoholic dementia are not uncommon. Alzheimer's disease is the common degenerative disease of the brain characterized mainly by premature and progressive dementia. The etiology is there is a severe loss of cholinergic neurons projecting from the basal forebrain to the neocortex, amygdala, and hippocampus, especially the fibers projecting from nucleus basalis of Mayonet are severely affected, which is also called as substantia innominata. Cerebral atrophy mainly involves the frontal, temporal, and parietal lobes. Pronounced neuronal loss occurs in hippocampus, entorhinal cortex, and parahippocampal gyri and subiculum. Pathology. Three pathologic features are the characteristic of this disease. That is presence of neurofibrillary tangles in the nerve cell cytoplasm is the cytopathologic hallmark of the disease. These tangles are fiber-like strands composed of hyperphosphorylated form of microtubular protein tau. They appear like pairs of helical filaments. The other characteristic feature is the appearance of neuritic plaques scattered throughout the cerebral cortex. The plaques contain amyloid protein, that is amyloid B protein or A beta protein, as the central core surrounded by the degenerating nerve terminal. Then there can be granulovacular degeneration of neurons, especially in the pyramidal layer of the hippocampus. Normally, amyloid precursor protein is secreted from the nerve cell. So this protein is hydrolyzed by the enzyme uh, gamma secretase to form A beta protein. This protein is hydrolyzed at three different sites by alpha secretase, beta secretase, and gamma secretase respectively. If APP is hydrolyzed by alpha secretase, non-toxic peptide products are produced. But if APP is hydrolyzed by beta secretase and gamma secretase, polypeptides with 42 amino acids are produced that are toxic in nature. The most toxic among them is A beta S 1 to 42, 142. These toxic polypeptides form extracellular aggregates that can bind to AMP receptors, and they also bind to calcium ion and increase calcium influx. They also induce inflammatory response and produce intracellular tangles. Eventually, the affected cell dies. Excessive and abnormal hydrolysis of APP results in more production of A beta proteins that form neuritic plaques. The neuritic plaques induce inflammatory reaction, tangle formation, oxidative damage, and neuronal degeneration, especially in the cholinergic neurons. The clinical features of Alzheimer's disease, usually the patient is above 50 years. There will be progressive development of the forgetfulness is the major symptom. The disease starts with the loss of short-term memory and later followed by loss of other cognitive functions. Dysnomia is forgetting the words, especially the names. Visuospatial disorientation, paranoia, and other personality changes usually occur. The Alzheimer's is treated mainly with the cerebral vasodilators, stimulants, and high dose of vitamin B, C, uh, and E have a beneficial effects. Trials of physostigmine, choline, lecithin, and cholinergic precursor and agonists have yielded some results. So this is uh, in brief about learning and memory and with some of the applied aspects related to that. Thank you.